Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everybody, to the Genetics Podcast. I'm really excited to be here today with Dr. Jonathon Hill, who's an associate professor at Brigham Young University, also known as BYU, and also the VP of Science and Technology and a co-founder at Wasatch Biolabs. It's a biotechnology that's focused on changing the future of early detection of disease, in particular with a focus on epigenetic testing and long-read sequencing that we're going to spend a lot of time talking about today. So first of all, thank you so much for joining us today.
Thank you, Patrick. I'm excited to be on today.
I understand that this is your second company that you co-founded. I'd love to learn a little bit more about the first one, but if we start with what you're doing today, if you could share the origin story a little bit. It's not often that people maintain an academic position and spin out a company. Often you're forced to choose, but it sounds like you've been able to keep a foot in both, and that can be really powerful as well to maintain those academic connections. Could you talk a little bit about the company and the origin of why you decided to start it in the first place and what you're focused on?
Yeah, happy to. And yeah, as you mentioned, I'm still a professor at BYU as well as the VP of Science and Technology at Wasatch. And we've really, I think, built a model where that can be really synergistic. We were born out of BYU. I have a colleague here who was working on methylation signatures and specifically looking at identifying cell types by using their unique methylation signatures, right? And he's mostly in the fertility space and using it for different fertility tests to look if there's sperm in the ejaculate, things like that, and diagnose different kinds of issues. But then he had a student that came to him who was an undergraduate at the time. And he said, wait, if you can identify these sperm cells, can't we identify neurons or other types of tissue in the cell-free DNA, this DNA that's just floating around in the blood? And they started to look into it and they thought this really could have major potential to look at neurodegenerative diseases, especially Alzheimer's, because in Alzheimer's and other neurodegenerative cases, you are losing cells, right? They're dying. And when they die, they release their DNA into the bloodstream.
Yeah.
And all of our DNA has the same sequence, but it has unique methylation signatures. So we can use those to identify the cell of origin, right? And so they first did a PCR bisulfite conversion based test for this. And they got really good results where they could separate out Alzheimer's patients from healthy individuals and thought, this could be a really great screening test as people become older. They could get a nice differential diagnosis and be able to start the emerging treatments that are coming out to hopefully extend,
Yeah.
their quality of life, et cetera. And then they ran into two issues. One, the PCR turned out to be somewhat finicky. It was only one locus. And when we talked to industry partners, they said, one locus is kind of scary. We want more, things like that. And the bisulfite conversion in PCR was creating quite a bit of noise in the system. And so they came to me, and I had been working on nanopore sequencing for a while. And they said, do you know anything that could help this? And I said, well, with nanopore sequencing, we can do native methylation calling on the entire genome, and we don't have to worry about either of these issues. And so we gave it a shot, and it really improved the results. And so then they said, okay, we're going to license this to a company, and get this out there. And they went.
Yeah.
And there was no nanopore-based sequencing companies out there doing clinical grade nanopore sequencing. It just was not available. And so they said, okay, if we're going to do this, we got to create our own company. And so they invited me. They said, hey, you're the nanopore expert. Why don't you come in and let's found this company together and create the first clinical grade, fully focused on nanopore sequencing company. And that's what we've created and built over the last couple of years.
Yeah, that's one of the big things that really caught my eye and wanted made me want to have the conversation because that was 2022, I think, when the company was officially founded. But even still today, there's very few companies that I'm aware of that do clinical grade long read, but it feels like the future to me. One of the special things about genetics is you can do it once and then you can read the data for the rest of somebody's life potentially. And long read
Right.
is in a way feels like the end game of genetics. If you can get kilobases of reads and you have confidence that you're seeing almost everything that there is to see, then it may be the last test you ever have to do in a clinical setting in some sense, right? So it feels like where it's all going to. Is that your perspective on it, or are there technology leaps that you think are coming beyond that?
Well, there's always technology leaps in the future, right? But
Yeah.
yes, I think that's where we're going. We've had Illumina style sequencing for 15 years now, and it's been great, and it's really helped kind of push genetics forward in the clinic. But there's always been limitations with short reads. First of all, you don't get a true telomere to telomere sequence out of short read sequencing. There's just some repetitive regions and other things that prevent that from working. With long read sequencing, we can get true telomere to telomere sequences, which gets us those hard to detect, hard to sequence genes. In fact, some people refer to it as the dark genome, this part of the genome that Illumina just can't capture very well. And we can get that part. And that's going to be very important for disease. We've also have reduced turnaround times and things like that that have been created uses where you really need to know the answer right now, right? Famous case that people like to talk about in the field is a case on the West Coast where a child came in and with a severe heart defect, and they were fading fast, kind of thing. And they needed to know if there was a particular mutation or not in order to effectively treat this patient. And they were able to use nanopore sequencing to create a super rapid telomere to telomere genome, identify the mutation, and get this kid the treatment that he needed, right? And so we start seeing these things we could not do before. The other thing that we're getting, you talk about, doing it once and then you're kind of finished. And for genetics diseases, that's often true. But there's also the epigenetic cases. And unlike your DNA sequence, the methylation changes or other modifications to your DNA change over time as you age. It changes in different tissues, it changes in different disease states, right? And so it's this dynamic thing that you really can track how those changes are occurring to create a novel biomarker that we've never had access to. And so that's another power that we have. And then finally, the last important thing, going back to the genetics, is long read sequencing gives you the ability to phase your mutations, meaning you can tell if two mutations that are separated by large distances in the genome are on the same strand, inherited from the same parent, or on
Yeah.
opposite strand. And so that's going to be a big benefit. And so, frankly, we were surprised when we went out there and nobody was doing it, right? There's a few labs that will have it. They tend to use it as orthogonal validation or kind of a side thing. But we're really the first lab to dive all in and say, this is the future. This is what we're going to do.
Yeah, I think that's great. One, if I take the position of the long read skeptic for a minute, something you hear a lot
Mhm.
is some people are even all in on exomes, right? Where they say, given the price point of an exome compared to a short read whole genome, if I have a fixed budget, I'd rather do an exome because I can diagnose most of the Mendelian cases. Obviously, if you're a family getting that testing, that's not a particularly useful answer. But going one step above that, you've got the whole genome using short read technology where you can use algorithms to piece together a lot of the information. When you think about that, like the trade-offs of cost and time and what you can find, what are the cases where you look at it today and you think actually like it's very clear we should be using long reads, but the community hasn't caught up to it yet for whatever reason? Because there's always going to be some cases where you want to use an exome, right? If you're doing new gene discovery in a research context, then maybe it's going to make sense there. But I'm interested in where's the edge where long read sequencing is really makes sense from a cost benefit analysis today.
Right. And there are costs, right? Right now, long read sequencing costs more per gigabase than short read sequencing. And I think for the foreseeable future, that's going to be true. And so it comes to these cases where short read really can't address effectively what you need. To give some examples, structural variants are much easier to detect with long read than it is with short read. Copy number variants, we get great results on. Um, Somatic mutations, methylation changes.
Yeah.
So it's when you need that data that you cannot get, right? Also, that dark genome really is an important area. There are many described clinically important mutations that exist in genes that just don't sequence well with the short genome.
Mm.
There's also a little secret here, and that is with adaptive sampling on the nanopore platform, we actually can produce kind of exome like
Right.
data quite inexpensively and quickly. And it's actually turned out to be really cool. We have a cancer partner who's interested in looking at tumor biopsies and sequencing those. And one thing we're looking at with them is doing adaptive sampling so we can get, 40x, 50x coverage in the exome, but we still get about 10x coverage everywhere else. And
Yeah.
so we've shown with them that we can detect structural variants very effectively in those kind of intervening areas. We can get low coverage methylation calls to look at epigenetic changes in the intergenic regions. And then we get that really good high resolution SNP coverage within the exons themselves. And so that creates a powerful data set that just isn't that accessible with other technologies.
Yeah, that's I want to talk about that a little bit just for everyone's benefit. If you're not familiar, nanopore sequencing is basically pulling DNA or RNA through a nanopore, and it detects changes in electrical current on either side of the pore effectively in real time. That helps you get rather than a visual readout, which is what you get from short read sequencing, right? It's fundamentally a fluorescence and imaging based technology. You're getting a electrical current and it helps you read the sequence with signal processing algorithms. Can you talk a little bit about adaptive sampling because it's really cool and it's really interesting, and I don't think many people are actually aware of it as an approach that you can do today.
Yeah, I mean, having that kind of different paradigm to sequencing is really where the magic is, because each pore is truly reading a single molecule.
Yeah.
And so, first of all, that's why we can get methylation data because we don't have to do bridge amplification or even library prep PCR and lose that methylation signal. But what you're talking about with adaptive sampling, what happens is as that read is coming through the pore, the machine is already recording those changes in the electrical signal. And so you're getting live base calling as it's being sequenced. It's not a post-processing kind
It's
of
very
thing.
cool, isn't it? It's amazing.
Yeah. And so it turns out within the first second, you can get about 400 bases of sequence. And the computers are fast enough now where they can actually take that sequence, align it to the genome, and mark it as region of interest or not region of interest.
Wow.
And if it's not region of interest, it actually reverses the polarity of that voltage and kicks the read back out. And if it is region of interest, it continues to sequence it. And so, it's not a complete exclusion of your non-regions of interest, but it's an enrichment of the areas that you're interested in. So you get something like 40, 50x within your regions of interest and a lower coverage everywhere else throughout the genome. And then using that data, you can make different kinds of calls on different regions.
Yeah, it's super interesting. How did you get interested in nanopores in the beginning from academic research? What attracted you to it?
Yeah, that's really funny you bring that up because you mentioned at the top of the podcast my first company, right? Pioneer Biolabs. And what we were looking at in that company was an enzymatic method for taking DNA or RNA from any species, any tissue, and using an enzymatic method to convert that DNA or RNA into a library of CRISPR guides. And so one use case that we tested, for example, my background is in heart development in the zebrafish. We could actually take out hearts from the zebrafish, normalize the cDNA from it, and make this library and target those genes that are expressed in the heart, not the entire genome, right? So it was a way to make this kind of semi-focused but still blinded library for CRISPR screening, for forward genetic screening. But we needed a way to sequence those libraries.
Right.
And because it's a library, um, we couldn't just do Sanger sequencing on it, right? We needed to be able to see each individual one. And we looked at Illumina sequencing, and for, the size and the kind of hairpins it made, it wasn't ideal. And then my student actually found a nanopore sequencer. The MinION nanopore sequencer is very small, very inexpensive. And he said, we can do this ourselves. We can take these libraries and sequence it on this machine. So this is 2018, 2019, and we got one and started working on it there and started picking up nanopore sequencing for that specific use case. And then it just kind of grew from there through the COVID years and everything since.
Yeah, very cool. You talked a little bit about Alzheimer's and some of the approaches to, for example, use methylation and cell type specific signatures to pick out neurodegeneration. Where are the areas of disease that you think are most amenable to this kind of early detection paradigm using either long read sequencing or pure methylation? I think you do a lot of standalone epigenetic sequencing as well. We've talked about,
Right.
in past episodes, we talked about circulating tumor DNA and other kinds of early
Hmm.
detection, newborn screening using genetics, other kinds of early detection paradigms. We haven't spent much time talking about epigenetics in the context of, broad disease surveillance. So I'm really interested to get your perspective on which areas are most exciting to you right now that developments are happening.
Oh man, there's so many. I mean, just within the kind of circulating cell-free DNA part, right? Which includes circulating tumor DNA. Um, so you've got this any kind of cell type of interest that you want to identify within the cell-free DNA. We can do it that way. And so that includes neurodegeneration, right? But it really includes any disease where cells are dying. So you can think of autoimmune diseases. You can think of, something we've wanted to look into is like cirrhosis of the liver that has a lot of cell death.
Yeah.
Other kinds of intestinal issues, gastrointestinal issues. We're actually looking at pathogen screening. Turns out that as your immune system responds to different diseases, it does so by setting up unique epigenetic patterns, and you can use that to identify certain diseases, or even in some cases predict outcomes. There was a really cool study a couple of years ago that took COVID patients who had really severe disease versus those who had more mild, and showed that there was an epigenetic signature that would actually predict that in the blood. Right.
Interesting.
We have partners who are interested in cancer and on many different angles, identifying cancer, identifying tissue of origin of the cancer. Because when you're looking at a metastatic cancer, sometimes it's hard to see where the original tumor came from. But of course, that's going to drive your treatment paradigm. We've got others, though, that are also looking at, hey, if I do radiation therapy on someone, I want to check two things. I want to check first to make sure the tumor is dying, but then I also want to check and make sure I didn't kill all the tissues around it, right? And so there's projects we're working on with those partners to try to identify those. So there's so many different ways. You really just have to think of methylation as this signature that tells you the state of the DNA at the time, right?
Yeah.
And so, uh, that can be disease, it can be tissue type, and it can be age. My
Right.
colleague that has a background here has done methylation clocks before, where you can tell the age of a person from it. So there's so many cool things that you can do, and you really can think of it as just a new class of biomarker that's out there, and we can apply it to so many different places.
Yeah, that's really interesting. How do you think about proteomics? It has been much more. I think it's burst on the scene the last couple of years, in particular Olink. And when you brought up aging clocks, for example, I think there was some pretty
Hmm.
interesting research last year on broad proteomic aging clocks, but also organ-specific aging clocks. Are these epigenetic signatures, do you think, likely to be complementary to proteomics? Or there's some areas where naturally is going to lend itself to proteomics because the proteins are showing up in the blood for whatever reason,
Right.
versus epigenetics? Curious to, I don't think you do proteomics today, but you probably messed around with it in the lab, I'm sure.
Yeah, we don't typically do it. I mean, proteomics is such a powerful tool because really the proteins are the things that are functioning,
Yeah.
right? And so to be able to test those and test, especially post-translational modifications, things like that directly is hugely powerful. They tend to be less stable. We have lower sensitivity in those tests, and the tests tend to be more expensive. So there's some trade-offs,
Mm.
right? And I see a future where there are cases where you're like proteomics are best, and there are cases where epigenetics is best, just depending on all those kinds of different factors, sample stability, price point, what you can detect, how sensitive it is, those kinds of things. Particular cases where I think proteomics will shine are in those post-translational modifications. I have a colleague here at BYU that works on redox changes to proteins, for example, right? You're only going to see that in the protein. There's nowhere
Right.
else that we can detect that. But then there's going to be others where, yeah, the protein change is what matters in disease, but you can read that by RNA sequencing or by epigenetic changes and use those as biomarkers to indicate that change in protein function. And then you've got a more stable sample that's easier to ship, easier to handle, and probably for the foreseeable future, cheaper to analyze.
Yeah, that makes sense. It's really interesting. When we before we I want to transition a little bit to talk about your academic role and the work that you do there, but I'm curious as we leave the long read sequencing world, are there I think you mentioned earlier that one clear area to use long read sequencing is when you've tried everything else and it fails, right? Which is actually very
Mhm.
often the case in rare disease, right? I spent a lot of my career working in rare neurodevelopmental disorders, and the diagnostic yield maybe 50% in some cases, right? So
Right.
it's half of families that get no answer even from whole genome sequencing. And so it's natural to try to understand what might be in parts of the genome that are hard to sequence or
Mhm.
combination or phasing issues that are difficult to detect. Are there any other areas besides like, almost a gene panel has failed, a whole genome has failed, and now we're in long reads? Are there areas where you're seeing people are going straight to long reads first? I think you mentioned rapid newborn sequencing is another great example. Are there any other examples where you're seeing like actually this is the first line of defense in the diagnostic pathway?
Well, I tell you, I think that in the future, especially as prices become more and more competitive, the answer is going to be just go long read because of what you're missing with the other ones. So you think about the cost of doing, an exome and then a whole genome with short read, and then doing, now you've piled all three of those. And we're
And the
getting to the point
time,
where it's
the time it
like, yeah.
takes. Yeah.
And we're getting to the point where you say, well, it might be a little more expensive than if the first one works, but the failure of the first one's so high that we really should just do everybody long read to start off with. And in the long run, we save money, right? So I think that's where things are going. It's just the prices have to come down just a little bit to get there, and also yields are increasing every day. Accuracy is increasing, those kinds of things. And so we're in that kind of technological stage where we like to refer to it like a wave, and you're on the
Yeah.
surfboard and you're kicking, and we're just about to stand up on this thing and go, and we're just trying to catch that wave at this
Yeah.
point.
I like it. It's a great metaphor. I was curious in how you balance the academic research, teaching, and also running and starting a company is hard. What does this split look like for you? I'm asking because a lot of our listeners have either are are in industry today, in academia. Many people are probably in both, and it's a common question for people to try to understand how do I make career decisions as well to stay in academia, to stay in industry, to
Right.
try to do both. And you manage to do both, it seems like, which is not all that common.
Yeah, it's really fun though. I really like it. It keeps my day exciting,
Yeah.
right? With more variety of things to do. But I think the key is that it's not that I have my professor job on one side and then the company on the other. We've really melded the two together. So this is how we've set it up. We've worked really closely with the research administration at BYU in order to do this. Everything's kind of above board. We're working with the technology transfer office, that kind of thing. But what we did is we developed the technology here at BYU, right? We were ready to commercialize it. We found there was no lab to do it, to license it to. So we decided to create our own, and we licensed it from the university. Now, a key to that is we had two kind of angel investors early on who said, we're going to take a risk on this. And then the student that had that original idea with the neurodegeneration disease said, I really think this is super interesting. My family has background in business. I've grown up around it. I want to take this and run with it. And so he became the CEO of the company, right? And spends his full time working on that. Licensed it from the university and kind of took it. But then what we did is we said, you know what, we need a research arm for this company. So let's create what's called a sponsored research agreement. Think of it as a company grant to the university. And let's fund research in mine and my colleagues' labs here at BYU. and have student projects that are working on some of these technologies and working on the improvements to the technologies we have. And then we don't have to create our own research arm. We get to build off the resources the university has, and we're going to continue to patent and license through the university, right? And so now we have this mutually beneficial thing where money is coming into the university, funding student projects, and creating technologies that the company can then license out of it. Now, the trade-off is, industry research, I've heard it described as uh, expensive and fast, and
Yeah.
academic research is cheap and slow, right? So there are some trade-offs there that we're trying to balance out. But one of the coolest things we found of it is by creating this neurodegenerative disease panel that we're now testing and kind of refining, we really created a platform that can be applied to many different use cases. And what we've had is we've had other students come to us and say, well, I think we could use this platform here, or I think we could use it there, and we let them go and we help them develop it. And then what's happened is we actually spin off companies where they become the CEOs of this company. And now they license with the lab to actually run their stuff, but they're
Yep.
taking care of the marketing and the all of that kind of work on it. And so we like to think that we're training biotech CEOs as undergraduates at our university now. And I think that's a really unique experience that most students cannot get anywhere, right?
Yeah, absolutely. And I think it also speaks volume to the from the university's perspective. It wasn't that long ago that a lot of universities were not particularly interested in entrepreneurship, right? Many of them, most of them, especially in the US, have changed their tune dramatically. But it used to be that there were
Mhm.
not that many universities where you could go and really choose to be an entrepreneur from day one, especially in when you get to doing a PhD. Certainly, I think a lot of PhD
Right.
supervisors had outward animosity towards their students pursuing anything that wasn't pure science. That's changed a lot in most places, but it's not like it's not 100% by any stretch.
It has changed. And I think one of the key things is, we've worked with the administration, and the administration at BYU as a religious institution has stated that their research goal is to create benefits for mankind, right? And so we've really been able to show them how this can create
Mhm.
impacts on society that they would not have in traditional academic research. And they've been flexible enough to say, okay, we'll allow you to pursue this. We publish a little less, we patent a little more kind of thing. And they've been flexible with that and allowed it. And so it's been really great to see and really great to see them work with us. The other thing they've done is they've realized that many universities have kind of put tech transfer rates, royalty payments, et cetera, a little too high to make it viable for startups to really take and run. And BYU has seen that and lowered some of those rates or taken equity in the companies, et cetera, in order to really help these things launch when they're not just being licensed by a traditional pharmaceutical company. And that's been a big benefit to us as well.
Yeah, I mean, how long have you been based in Utah? There's a long tradition of great human genetics in Utah as well. I remember when I was doing my PhD at the Sanger Institute, I would read a lot of the papers that came out of Aaron Quinlan's group, for example.
hmm
Did you grow up in Utah, or have you been there? You've been there for more than 10 or 15 years, I think, right?
I've been here a while now, but I grew up in the Silicon Valley in
Oh,
California, and
interesting.
so my dad was a programmer until he recently retired, and I grew up around that kind of environment with startups all over the place, the kind of growth, the kind of craziness. San Jose in the 80s and 90s, essentially, right?
Mhm.
So it was prime tech time. In fact, my grandpa worked for IBM. He designed some of the first computers for
Huh.
IBM back in the 50s and 60s. So it's been in my background for a long time. And now I see the kind of excitement and growth we had in the 90s in the Silicon Valley. I see that happening in biotech right now. And I think it's a real opportunity for lots of people to get into and really help drive this forward.
Yeah, that's great, and it's interesting the way that you've used the company as a launch pad for so many other companies. That's pretty unique actually, and it helps to create a little bit of an ecosystem around the university and the company and technology, right? It's a good way to get those win-wins.
It is, and we're trying to build the ecosystem here. That helps everybody. If you can create that kind of workforce and ecosystem where it draws talent to the area, things like that. Although Utah has one of the biggest biotech sectors in the United States, it's kind of this hidden gem in the middle of the
Yeah.
United States. Most is on the coast, but we've got this little island here where a lot is happening, and it's an
Yeah, I
exciting
believe
time.
that. Why do you think that is? What is it that is well set up there?
I think partly, Utah's a very business friendly state, first of all, which really helps, especially in these kind of high risk ventures to have that.
Yeah.
But also, I think it comes back to what you were talking about, Aaron Quinlan and others doing this great genetics work out of the University of Utah mainly, and now more and more out of BYU. And creating that. We have some unique things here like the Utah Population Database, and resources like that that have really helped kind of launch those things. And you've seen mainly genetics companies like Myriad Genetics and things
Yeah.
like that come out of that kind of environment.
Yeah. I was curious about the name. Where does it come from? Is there a story behind the name Wasatch Biolabs?
Yeah, so Utah, most of the cities are along this nice mountain range that extends right through the middle of the state, right? And it's where all the ski resorts are. So where the
Right.
Olympics were held when they did it, et cetera. That is the Wasatch Mountain Range.
Ah,
And so that's what we named it
there
after,
we
is the
go.
mountains right behind us. It's a Native American word that means mountain pass. And so they named the mountains and we named the lab after the mountains.
Very cool. I like that. As we wrap up here, I'm curious to learn about any technologies or things that you have your eye on that you think maybe people are underestimating or not fully appreciating the benefit of it. I get the perspective that based on your role and the broader research work that you do, that you probably keep your finger on the pulse of lots of new and interesting things that may not be ready for prime time yet, but could be in the next couple of years. So besides epigenetics and long reads that we talked about, are there any other areas that you find are particularly interesting?
I mean, those are the two big ones, but really where I'm excited, and this is going to sound a little cliche because perhaps it is, but I'm really excited to see how AI matures in the
Yeah,
diagnostics space. There's a really interesting paper that came out of, I think it was UCLA recently, where they took complicated cases and they gave them to doctors, and then they ran it through a large language model that wasn't even purpose designed. And the large language model far outperforms the doctors in accurate diagnosis of these diseases, right? And I look at it, and my work in the diagnostic space just tells me that no disease is simple. It's a
yeah,
complicated pattern of changes. And some of these are going to be epigenetic, they might be genetic, they might be proteomic, right? You've got all these data points, and combining that is just purpose made for machine learning and AI. Now, the key, I think, and what we need to do to really make that effective moving forward is think about those inputs into the AI, right? How do we gather the data in a way where we have low noise, we have clean signals, so that the AI can parse through all of that data and learn from it, right? And make a good diagnosis. And I don't think there's enough attention on the inputs. There's a lot of emphasis on the models themselves using existing inputs, but I think we need to be thinking more about how do we create the data that will make it really effective.
yeah. I think that's a really good point. And I have similarly been watching the various benchmarks, both medical and otherwise, over the last couple of years. There's a interesting, this is not directly related to medicine recently, but I listened to a podcast where someone was talking about a benchmark that is designed to test general intelligence, effectively large language models. I think it's called ARC, ARC. And the thesis of the people who designed the benchmark was that LLMs are fundamentally memorization machines. And
Hmm.
one of the things that makes us as humans unique is that we can encounter the unknown and adapt and
Right.
pull things that are out of the distribution. And so they designed this very clever benchmark that is very easy for humans to pass. Actually, most humans get 70 or 80% on it. But LLMs were failing miserably because it was fundamentally not solvable by rote memorization, but required
Hmm.
something else, some kind of problem solving. But then OpenAI's latest model just got 77% on it or something. So
Wow.
it's like, wow, it's they made this like very good and very hard to crack benchmark, but then the latest model just got
Just
on
did
par
it.
with a
Yeah.
typical human for this thing that was supposed to be only really achievable by something demonstrating the kind of general problem solving abilities that humans have. So it doesn't mean it's going to solve medicine overnight, but I have just been continuously amazed at how like each benchmark it's hitting the best possible
Yeah.
scores on the MCAT, right, or other kinds.
Right.
And that's not
Yeah. I
medicine in
mean,
the real world, but it is something.
yeah, and I think, in my mind, I think of AI as just a really good complex pattern recognizer, right? That's
Yeah.
what they do really well. Outside of the large language models, more of your traditional perceptrons and things like that. So they look at patterns and they see patterns that are hard for us to detect because the data is so big. And that's medicine's just built for that, right? Because that's what
Yeah.
medicine is a huge complex pattern. You got to put all these pieces together and make a call out of that. And so it's really fit. I do think we're in somewhat of a bubble in the area. It's a buzzword, everybody's using AI. Not there's
Yeah.
honestly cases where I see it and I'm like, you don't need AI there. There's a much simpler
Yeah.
model you could be using. Um, and people call things AI that aren't really right. So the field is young, and we're going to see some of that shake out and some uses become not so great. But I think there's going to be some gems in there that are going to be really awesome.
Yeah, I completely agree. Well, Jonathan, thank you. I really appreciate you joining us, and congratulations on all the great work that you all been doing.
Thanks, and thanks for having me on. It's been fun to talk to you.
That's my pleasure. Thanks everybody, as always, for listening, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.